Hey, welcome everyone. My name is Samir Velankar. I'm team leader for PDB team. It's my great pleasure to welcome you all for the 2024 PDB Art exhibition opening event. This is our 9th year. And I want to really congratulate the team behind this whole effort. So Deepti coordinates the whole project, but she gets help from many different teams at EBI. So I want to thank them all. And it's not just that we are running it for 9 years. I think it has been growing for 9 years as well. We started with one school and one student 9 years ago. And now it's not just in Cambridge, but we have students in China, in Australia, and recently in many other European countries participating in this. So you have peers in many different countries who are doing very similar efforts as you all. So just to say a little bit about EMBL and the European Bioinformatics Institute, which is the place we are at. So this is the place where we host all the data for life sciences. So you can think of it as a library, a digital library of all the biological data. And many researchers, millions of researchers use it every day to take this data and drive their research. And that includes maybe drug discovery. It might include addressing climate change, etc. So we have data on genes, on proteins, on small molecules. So every type of data you can think of, we host it here, and we then openly serve it to the whole world. So this data is completely free for anyone to use. And that is our mission. Our mission is to keep this data open and drive research. So one of the things we constantly think about is how do we present this data? And data visualization is quite key to using this very complex data that we host. And in developing these visualizations, as you will see later, it's not just about putting that data on the website. It's about thinking about how people will interact with this data, how they can understand what's the knowledge that is in that data. So apart from running this project here for 9 years, EMBL has been around now for 50 years, and we are celebrating our 50th anniversary. And as part of that, this project was adopted by EMBL in various engagement events, and we have contacted many European countries and schools there, and who are now participating in a competition that is being hosted by EMBL. And Deepti will tell you a bit later how to participate in that competition if you are interested. So finally, I think as you all know, you have seen really amazing biological data, so really beautiful macromolecules, and we are lucky that we host this type of data and provide it to the whole world. And so this project, PDB Art project, really helps us share that beauty, that real knowledge that we have in the data with everyone. So thank you again for coming here, and I'll pass it on now to Deepti, and we'll enjoy the rest of the opening event.
Thank you, Samir. Thank you everyone for joining. And as Samir mentioned, I am Deepti Gupta. I lead the PDB Art project, and I have been working with the schools and students for 9 years now. And it is a delightful project, and I want to welcome you all for this opening event for opening the PDB Art exhibition 2024. So thank you for joining both online and in Kenru Lecture Theatre on campus. I want to remind everyone that this event is recorded and it will be shared afterwards. So this is the brief outline of the program. In order to know more about the project, the best way to show you is through the guided tour. This will give you a glimpse of the artworks that were submitted for this year's exhibition. And all of the artworks, if not all, most of it is on the online exhibition. Welcome to the PDB Art Exhibition 2024. The PDB Art Project is a global outreach initiative that brings together schools, art societies, and the Protein Data Bank in Europe to spark creativity and curiosity in students aged 11 to 17 years. Through this project, students dive into the fascinating world of 3D molecular structures in the PDB, transforming their discoveries into inspiring works of art as part of their curriculum. This exhibition celebrates their unique creations, offering a glimpse into the imagination and talent of these young artists. Today we invite you to journey through their creations, where each piece tells a unique story of curiosity, imagination, and science. Our first stop introduces the artwork from Xiong Xiu, age 13, a student at Tsinghua International School from Beijing, China. From a very young age, he has been fascinated by animals and has chosen to explore the cardiotoxin protein from the venom of Chinese cobras. In his artwork, he captures the tension between danger and healing. He uses a red and green color scheme to symbolize these themes. His comic book-style artwork features 3 characters: the snake, the horse, and the human. Using watercolors and acrylics, he creates imagery that highlights the complex relationship between peril and hope. The horse represents the vital role of animals in producing antivenom, merging artistic expression with scientific themes. We next explored the artwork based on enzyme anthocyanidin synthase that is responsible for producing vibrant colors in flowers. Here, Hobin Li from Tsinghua International School, Beijing, China, was inspired by this fascinating enzyme and the delicate blue colors of the flowers. Hyobin combined scientific curiosity with artistic exploration by using fluid art to depict iris flowers on a bench and incorporating cyanotype to illustrate the 3D protein structures of this enzyme on the backrest. This piece creates a captivating blend of art, science, and nature, transforming everyday objects into functional art and inviting viewers to engage with it. Moving forward, here are artworks created by Year 12 students of Saffron Walden County High School in the UK. On the right, Louis Longarti's thoughtful creation is inspired by hemoglobin, an essential protein that carries oxygen throughout the body, highlighting its crucial role in blood system and its connection to the heart. The artwork uses pen strokes and ink techniques to capture the flow and movement of hemoglobin. To the left is Evie Brown's artwork that takes inspiration from the protein BDNF, that stands for brain-derived neurotrophic factor, a protein that is crucial for brain plasticity, learning, and memory. Fascinated by how BDNF levels increase with exercise, enhancing cognitive function, Evie uses marbling ink and dry point prints to depict the brain's intricate fluid structures. Next, and all along these walls, are artworks created by Year 8 students of Thomas Gainsborough School in Sudbury, UK. They explored proteins from various animals like cats, donkeys, fish, and chicken. Here, on the left, Magnus Pringle Green and Lucy Lee on right tackle 2 urgent global issues: antibiotic resistance and environmental degradation, respectively. Magnus's cat-inspired artwork sheds light on the hidden transmission of antibiotic-resistance bacteria or allergens from pets to humans, urging better hygiene and awareness. Meanwhile, Lucy's artwork on the right portrays endangered mountain ecosystems, symbolized through the hemoglobin structure of donkey, and calls for greater care and protection of our planet's fragile environments. This piece is inspired by the P53 protein. In this artwork by Yunan Liu from Tsinghua International School, China, P53 is portrayed as a vigilant guardian. P53 is a vital tumor suppressor protein that regulates the cell cycle, facilitates DNA repair, and induces apoptosis in damaged cells, playing a key role in cancer prevention. Nan Liu's imaginative piece beautifully transforms P53's protective functions into a stunning visual narrative. The bow and arrow motif, highlighted in yellow and purple, symbolizes P53's tumor-suppressing role. The pleated skirt design is adorned with DNA chains crafted from silk mesh and rice beads, emphasizing the protein's DNA binding domain. This artistic interpretation underscores the crucial role of P53 that it plays in safeguarding human health. Next, William James, aged 13, from Warrandyte High School, Australia, explores the impact of nicotine in his artwork on the right. His sculpture, crafted with clay and acrylics, represent the addictive grip of tobacco. The nicotine in tobacco binds to nicotine acetylcholine receptors in the brain, triggering psychoactive effects and contributing to over 7 million deaths annually, making it the leading cause of preventable death worldwide. Inspired by a fleeting encounter with a smoker, William captures the dangerous allure of nicotine and its devastating health impacts. In a parallel exploration, Tessa Slater and Xing Zhongxiang focus on yeast called Pitrosporum, which are naturally occurring on human skin and part of skin's normal microbiome. While generally harmless, these yeasts can trigger immune response when they invade the outer skin layer, leading to conditions such as ectopic dermatitis and eczema. This artwork depicts a hand emerging from the canvas, showcasing visible dermatitis and the challenges of living with this condition. Together, these two pieces prompt reflection on the health crisis of nicotine addiction and skin diseases, shedding light on their significant impacts. Spidroin is a silk protein produced by spiders, known for its exceptional strength and elasticity. Here, the artist Zi Mo from Tsinghua International School, China, was fascinated by its potential across various fields and explored its unique transition from liquid to solid, which inspired their artwork. She meticulously crafted the structure of spider silk protein using a 3D printing pen, with wool cleverly mimicking spider silk to make the protein as close to a spider's web as possible. The branches in the installation simulate a natural environment, creating an atmosphere that makes viewers feel as if they are deep within the forest. Our final stop brings us to Maddie O'Connor's artwork, a student from Stephen Peirce Foundation in Cambridge, UK. Her piece is inspired by the AlphaFold predicted structure of pea protein, which plays a key role in human skin and eye pigmentation. Maddie selected this structure from the AlphaFold database and skillfully combined watercolors and digital techniques to capture the elegance of this protein. This marks the first AlphaFold-inspired artwork in our collection. With this, we come to the end of the guided tour. Thank you for joining us on this journey. Each piece in this exhibition showcases the curiosity and imagination of the students that is guided by their teachers. Together, they highlight the remarkable connections between art, biology, and the future of scientific discovery. We hope you leave feeling inspired. Please take your time to explore this wonderful exhibition further. Thank you so much. I would now like to introduce our speaker, Veronica. And we are delighted to welcome Veronica Falconeri Hayes. She is a certified medical illustrator and animator with over a decade of experience in creating compelling cell and molecular visuals. As a founder of Falconary Visuals, based in Washington, D.C., Veronica specializes in developing innovative visual solutions for science communications challenges in pharma, biotech, and academia. She holds a degree in Master of Arts in Medical and Biological Illustration from Johns Hopkins University and has collaborated with prestigious institutions like NIH. Her expertise spans concept development, illustration, animation, and molecular model building. Veronica is currently certified by the Board of Certification of Medical Illustrators with her credentials valid through December 2026. I will now pass on to Veronica to tell us more about what she does.
Thank you for the introduction. I'll pull up my slides. Okay, so as Deepti said, I'm Veronica Falconary Hayes. I am the principal of Falconary Visuals, and I do molecular illustration day in and day out. I've been doing it for a while now. And first I would say I really enjoyed seeing the exhibition, and so I got to take a sneak peek last week and go through the 3D version, and I really, really enjoyed seeing all of the students' takes, kind of the fresh eyes that you have on molecular structure. I've been doing it for over a decade now, and I kind of have a style, and I feel like I kind of have this narrow focus, and it was really refreshing to see just all those fresh takes on molecular structure, and I think really remind me just how much all these little molecular structures affect our larger lives. And I really appreciated seeing that connection between this tiny world of the molecular and the big world of health and public health and global health as well. So I really want to commend you guys on doing really amazing, inspiring artworks. So I'm going to give you guys a little bit of a career overview today, so a visual career overview, hopefully. So first, just kind of a quick review. So what I do now is I'm a freelance certified medical illustrator and an animator as well. So that means I get to make visuals explaining science, mostly biological, day in and day out for a lot of different clients. I am a certified medical illustrator. I'm a member of the Association of Medical Illustrators, which is a US-based or a North America-based organization. So if you are interested in learning more about medical illustration, that is a good place to go. So how I got there, first I got my Bachelor of Arts in Biological Science at Smith College. I was an art minor at the time. And then I went on to get my Master of Arts in medical illustration specifically. In North America, there are 5 programs for that. There are also programs in Europe for medical illustration. I'm not as familiar with those, but I just wanted to let you guys know. So a lot of my information is based on the education system in North America, but there are similar programs in Europe. After I got my master's in medical illustration, I actually worked within a research lab for a few years. And that research lab was at the National Cancer Institute here in the US. And they did cryo-EM. So these structures that you were looking at, you might have learned a little bit about where they come from. So scientists use different techniques to figure out the structures of proteins. Most of the ones in the PDB right now are still from X-ray crystallography, which is, a really good technique to determine the structures of proteins. Some are from NMR or nuclear magnetic resonance, and then a growing portion are from cryo-electron microscopy, which is another way to determine protein structures. So I was actually in a lab where scientists were using cryo-electron microscopy to figure out protein structures. So I got a really deep dive into molecular structure and structural biology at that point. And that's kind of defines the path of my career, I would say. I was always interested in molecules, and that really gave me the deep dive that I needed to continue to pull that through my art for the rest of my career. So I want to give you guys sort of a visual look at the different art and kind of the development of my art and science knowledge from age 18 onward. So this is even a little bit older than where you guys are now. So as an undergraduate, first I really fell in love with cells and tissues. So here are these are histology slides that I created in undergraduate. So these aren't drawings. This is actually slices of tissue that I stained with particular chemicals in one of my classes to create contrast so I can look at different cellular structures. So on the left-hand side, you're actually looking at a slice of brain tissue from a mouse. And you can see some of the neurons and all the neuropil, all the axons sort of interdigitating in dendrites. And on the right, that's actually a cross-section of the esophagus. So, I was really falling in love just with like the intricacies of cellular and tissue structure at the time. And I really, really enjoyed doing this histology class because I got to, prepare all of these and then look at them under a microscope and kind of spend time, trying to take the best image, but also just sort of soaking in kind of the organic feel of everything and starting to understand how everything worked on a very microscopic level. But this is still much larger than molecular, of course. At the same time, I was also able to do an independent research project where I did immunofluorescence labeling, which you can see some of that here. And I was actually growing cells in a petri dish, and they were little muscle cells actually, and I would grow them and then try to figure out where different proteins were during different stages of their growth. And then, so that was all my biology work. And then I was also, of course, developing my art skills. So these are some of the drawings that I did as an undergraduate. So some of them, like on the left, you see the stairs that I was actually sitting somewhere drawing what I saw. Some I was studying from art, and then some were completely creative. Like you've seen this, bizarre panda scene on the bottom right. Then right after undergraduate, I went straight to graduate school, and I'll show you some of the work that I did there. So I started developing more specific medical illustration skills. So each class that we had there was a combination of learning a new media technique and then also learning new science. So this was actually for our surgical illustration class. So we had this class where we would actually go into the operating rooms at the hospital there, and we would observe the surgeries. And we'd sketch, and then we'd take our sketches back, and then we'd compare it to our other resources, our textbooks, our knowledge of anatomy, and create these illustrations to summarize the key points of the surgery. This was I think my very first 3D molecular illustration. I was still learning how to use the 3D software. Now I look back at it and I'm like, oh no. But at the time I was pretty happy with it. But, obviously I've grown so much since doing this piece. So it's always fun to look back. We also did more general science illustration. So here you can see this is an illustration of how frogs take care of their baby tadpoles, and we were working with the National Aquarium for that. That was a fun project. Here's, again, one of my first forays into 3D software where I was illustrating a neuromuscular junction where the nerve comes into to synapse or to interface with the muscle. And then after graduate school, I went, as I said, to the National Cancer Institute's high-res electron microscopy lab and started doing a lot of molecular structure work. So here are a lot of those pictures. So I did some editorial work, and I think this is maybe the stuff that's closest to, kind of the work that you've done. So a more artistic take on some of the structures. But I also did so many figures for publications where the scientists were explaining exactly how these little molecular machines worked. So lots and lots of detailed schematics. And I'm sure now that you guys have done a little bit of work with the molecular structures, you're starting to recognize, oh yeah, that's like a ribbon diagram. I know what that is. These are all based on PDBs. So yeah, I have to say it's amazing having the protein data bank and EMBL doing all of that work to keep that data up, that's a huge help for when I'm creating an illustration like this, to just have all of this data right at my fingertips to be able to pull these structures and just download them. At the same time, I started to do a little bit of freelance work on the side while I was at the NCI. So this was actually my first illustration for Scientific American, which is a publication here in the US that is for more general audiences, but they they have articles about scientific topics. And this one was about HIV. And this illustration was showing how HIV has a spike on its surface and it connects to immune cells. So I was using structures from the protein data bank and to show that spike, to show the different pieces, and to show the receptors on the immune cell surface that it attached to. So after a while working in the high-res EM lab, I knew I wasn't really growing as an artist so much anymore. I wasn't learning many new things. So I was like, okay, like what can I do next to help myself continue learning, to help myself continue growing as an artist? And I had always wanted to try going freelance so that I could kind of have a greater diversity of projects. So in the high-res EM lab, I was doing a lot of scientist-to-scientist communication. It was a very narrow focus. But the nice thing about going freelance is I now have a much broader focus, and there are a lot of different people that I work with. So I still work with researchers and scientists, but I also get to work with biotech companies, publications to an extent, and that diversity is really fun for me. So these are some cover designs that I've done. Some of them are very structure-based. You see on the left the cell reports methods covers that I worked with a structural biologist who determined the structures that you're seeing on the cover. And we really wanted to highlight, exactly what the structure that they were publishing about. Some are a little more metaphorical, as you can see in the middle, the Science Translational Medicine cover. We've got these little almost like rubber ball viruses bouncing off this metal shield of antibodies. And some are like a little bit in between, mixing like a cellular scene with the molecular scene. Here's some more of the covers I've done. Because I have this sort of fluency in science, I do not just biological illustrations sometimes. Sometimes I will like cross over into material science, which you can see with that middle cover. Just because the ability to talk to scientists that I've developed over my career, I'm able to use not just for scientists in biology, but also for scientists in other subjects, and to help them translate their science into visuals. On the right-hand side, you can see one of my more well-known covers, which was the cover for Scientific American in July 2020. Because I just had a lot of experience illustrating viruses and such, at that point, of course, when COVID hit, I was kind of leading the field a little bit with my illustrations. So that ended up being a good opportunity for me to get that cover where I was able to put together this illustration of the coronavirus at that point. So I also do 2D work. So some of the work you're seeing here, this is all done in almost all done in my 3D program of choice, but I also do sort of more flat work. This is in Adobe Illustrator. It's vector artwork. Similar programs are like Inkscape, that kind of thing. So this is for a publication summarizing, a mechanism or how outer membrane vesicles bud off from bacteria. This is more a general immune system illustrations explaining how it works. And you can see these have just a different feel than the 3D artwork. Sometimes I get to mix 2D and 3D. So this is, again, another piece for Scientific American, explaining how DNA and chromatin specifically gets organized in the nucleus without getting tangled. So on the left side, I did sort of this engaging 3D look at like all that chromatin, a little simulation of maybe like how the chromatin would pack into the nucleus. And on the right-hand side, I used, again, a more simplified 2D approach to show the actual mechanism of, there are certain proteins that sort of extrude it into loops to help it stay organized. And this is, again, another, you can see some of the molecular structures here, but it's more of a 2D approach showing how CRISPR works and in some ways that it might be applied. But you can see there's like a little CRISPR molecular structure in there. And that's, again, based on a PDB structure. Here's, again, something that mixes 2D and 3D. And you can see on the left-hand side, that is a structure from the PDB, of course, from the protein data bank. And so I was able to show that in 3D, show where certain mutations were. So this is actually the spike from the coronavirus protein showing the mutations in the Delta variant. And on the right-hand side, you have a 2D illustration that explains how that affects the viral life cycle. So, how does it change how it attaches? How does it change how it gets prepared, how the new viruses get prepared, that kind of thing. So I also do general anatomy work a little bit. So you can see here again, I'm mixing a bit of 3D and 2D, some different anatomy illustrations, looking at periodontitis in this case, so inflammation around the gums. I get to do a lot of different things and I really love that. Here's something that is, again, more structure-based. So this was for the Ewers lab for one of their publications. And a lot of times the cool thing is I get to work with structures before they're published in the PDB. So I'm working with the scientists who have created them, or have determined them and built the models. So I get to then work with their PDBs and create their illustrations to go with the papers that get published along with the structures. Here is some work that's actually for a biotech company. So they have a technology that targets cancer cells. So the antibody targets cancer cells, and then they have a specific little radionuclide that comes and kills the cancer cell. So here you're seeing PDB structures for the antibody and for the receptor on the surface that it's binding to. And here's another one for a biotech company, and this is lots and lots and lots of PDB structures, trying to look inside a cell with molecular detail and show what's happening in a heart cell in heart failure and how their therapeutic can potentially help that. And finally, I do some animation, so I'll just let you guys watch. This is my sample reel. It has lots of little clips of larger animations that I've done. And finally, I want to finish with some art that I did back when I was about the age of the artist in this exhibition, because I really want to reinforce that art takes a lot of practice. So here's something that I was doing at age 15 in Microsoft Paint. So that was the digital media available to me at the time. I shudder to think how long it took me to make these things. Here's a little cow scientist. I forget what I was thinking. It was a while ago. I think I had a whole series of different kinds of cartoon cows. And then this one, so I drew a lot of them by hand, and for some reason this one ranked and it got to get drawn with a mouse in Microsoft Paint. Here's some of my pencil work at the same time. So this was the art that I was making for fun when I was in high school. And I obviously like my skills have increased since then, and I've also like learned lots of different media, but I guess it's been almost 20 years since I made some of these. So if you are thinking about a career in any sort of art or design or that kind of thing, and you're like, oh gosh, I'm like never going to be that good or whatever, I just want to put this in there for reference, just so if you're looking at the work that I do now, this is what I was doing in high school for fun. Like I was doing some still lifes and stuff in art class, but just let's have this as a reference point. So that is all I have for today. Again, wonderful to see the exhibition, and thank you for listening.
Thank you so much. Thank you so much, Veronica. That was a wonderful, wonderful talk. I thoroughly enjoyed your work over the years, and I can see there will be lots of questions for you. So people online can put their questions in the Q&A box, and we will put them through to Veronica and Lucy in the Q&A session. Okay, so moving on, I would now like to introduce Lucy. Lucy Walker is a PhD student in chemistry and physics at the University of Cambridge. She is researching the design and synthesis of organic radical molecules that can absorb and emit light. Alongside her studies, she creates pointillism-inspired art using skeletal molecular structures, which she sells through her Etsy shop called Molecule Arts. Her artworks depict everyday objects composed of molecules found in them, such as her piece of bee made up of molecules present in honey. Lucy's passion for combining art and science began during lockdown, leading her to Open Molecular Arts, where she has now sold over 700 prints worldwide. She is here to talk to you about her science and art journey and how she combined them to create her business. So welcome, Lucy.
Hi, I'm Lucy. I'm a second-ish year PhD student at the University of Cambridge. I say the ish because my third year actually starts tomorrow, which is quite scary. And I study both chemistry and physics. And on the side, I'm a science artist and illustrator. So the science art overlap, I feel like for some reason it's kind of embedded in us that science and arts cannot be related. They're different subjects at school. And I think there's a lot more overlap than you might think. So I'm giving you just a few brief examples, which were actually nicely introduced in the previous presentation. So one example of places you can find science art is in scientific journals. And if you don't know what one of these is, they're kind of like magazines for science. And this is where scientists will publish their most up-to-date, state-of-the-art findings for everyone to read. So artists are used to create captivating images for the covers to entice more people to read science. Also, visual media. I think this gets forgotten a lot, film and TV. Both of these examples on the screen had actual scientists working in the production crew to make sure the science within the film or TV program was as correct as possible. Then figures, this is kind of also what Veronica was talking about, and these are found within the scientific journals. And I would say this is the primary way that scientists will get across their research ideas, because it's much easier to have just a single image of the scientific discovery than having to read paragraphs and paragraphs of text. So having an artistic mind to develop these really helps to make them concise and easy to follow. And then finally, commercial. This is where my art business kind of fits in. And many people love science gifts. And so science artists can develop the gifts they would like. So why am I standing here and talking to you? Well, I thought I'd give you maybe a brief summary of my science and art journeys, which for much of my life have been very separate until the last couple of years. So what was I studying? I started secondary school in 2011, which, yes, is quite a long time ago now. And I did my GCSEs in 2016. my A-levels in 2018, which was followed by going to university to do my master's degree in 2018 till 2022. And then since then, I have been doing my PhD. What was I drawing at these times? Well, for the first 13 years of my life, I pretty much exclusively drew horses. Yes, I was a horse girly, and that was very much reflected in my art. This somehow transitioned into portraits, and not portraits of horses. And I did that for many years between secondary school and sixth form, which then naturally transitioned into a photorealism phase, pretty much starting during lockdown in 2020. And if you've not come across photorealism yet, that's basically you have a photo and you're trying to replicate that as closely as you possibly can using various different media, but for me, that was colored pencils. And then towards the latter end of COVID until now, I pretty much exclusively do science art, but I do it in various forms, which I will go into during this presentation. But anyway, time for a trip down memory lane. I thought I'd give you some evidence of these eras I went through. So here is a small selection, and like this is probably like 1% of the horses I ever drew. And so, yeah, you can see these, and then you can see me in the bottom left, probably about 5 or 6, being very abstract at my easel. Then we go to secondary school. This is when I was going through my portrait era, and I was obsessed with drawing things of people I like or whatever. So I've only got one picture of Taylor Swift on there, but there was a lot of Taylor Swift, but I thought I'd spare you seeing many images of her. And I've listed my GCSEs on the board, and you will notice a distinct lack of GCSE art, which was not what I wanted. However, at my school, you have to choose between triple science and art because of the option blocks. So unfortunately, I had to sacrifice art because I thought science would be maybe more useful for my future, which is a good job I did it, considering now I'm still doing science, like over 10 years later. So yeah, that was my secondary school phase. Then sixth form, I didn't have much time to do a lot of art during sixth form because A-levels was quite intense, but I was kind of transitioning between my portrait and photorealism phase. I've listed my A-levels on the bottom. The pink post-it note is where they started, and the blue post-it note is what I actually ended up getting. And you'll also notice lack of art, and that's because despite I had moved schools, they would not let me do A-level art without having done GCSE. So yes, unfortunately, my art training had ended in year 9, which was actually very upsetting to me. Anyway, so my A-levels changed. Basically, what happened was I did not know what I wanted to do with my life coming out of secondary school. And so I thought, let's just pick A-levels I'm going to be maybe okay at. So that's why I ended up with chemistry, DT, geography, and PE. At the end of my first year of A-levels, I quickly realized that chemistry was what I wanted to do with my life. Then my school took me to a university, I guess, careers fair. And every single university I went to speak to said they would not take me for a chemistry degree because I had not done another science or maths as A-level. This caused me to spiral into a frenzy, which resulted in me having a life crisis and going to my school and begging them to let me take A-level physics in a year. Somehow I got them to agree, and somehow I managed to get the A-level, which was very worth the hustle because it allowed me to do my degree of my dreams. So for those of you who are thinking of A-levels soon or future plans, like, don't stress too much over your options. With a lot of hard work, you can still do what you would like. And yeah, anything is possible. So after the A-level stress, I finally got to start my master's degree at the University of Birmingham, and I had the time of my life there. And so I did something called an integrated master's degree. I feel like it's probably the less conventional degree type. Usually people would do a 3-year bachelor's program, graduate, and then do a 1 or 2 year master's on the end of that. Whereas I did 4 years, no graduating, got the master's. And for me, the reason I decided to do that was because I just wanted to do as much science as possible in a short amount of time. And I think it helps with funding as well if you do an integrated master's. So yeah, that was my choice for that degree program. I've listed scholarships ! On the bottom because I think a lot of people don't realize when applying to universities, they have lots of scholarship programs going on, which can really financially help you out as a student because student life is very tough on student loans. And I did quite well out of this, and it really, really helped me to afford living and all of that at university. So yeah, some photos on the screen. On the far left, that's me holding up my master's report, which is like a 12,000 word summary of what you've done in your final year at university. And on the right, you can see me looking very proud of myself in the lab. Not quite sure what I discovered there, probably nothing. So unfortunately, despite I absolutely loved my degree, COVID hit right in the middle of it. And that was quite upsetting because it meant between my second and my fourth year, I had to go back home, which was lovely. I really like my parents and living with them, but when you're out trying to live your student life, going back home when you're 20 is not what you wanted. However, I tried to make the most of it. And one thing that I fell back in love with was art, because I hadn't had much time to do any at university so far. So I really hit my photorealism era. I invested in some new colored pencils, which really helped me. And yeah, I spent all of my time doing photorealistic drawings, or as close as I could get. I'm aware they're still not perfect. So I've listed some of my favorite ones on the screen. My particular favorite is probably the snake, because it's all cut out of one piece of paper. And it's essentially a trick on the eye with shading and light and camera angles to try and make it look realistic. So I was very surprised at how well that came out, because I was expecting it to look like it had been flattened by a truck or something. And then I've also put the Adobe Photoshop logo in the bottom right. When I kind of run out of things to do and wanted something else to occupy myself with, my dad was like, oh, why don't you just learn Adobe Photoshop? It might be useful. So I thought, okay, well, I'll learn this. It probably won't ever be useful for me. And so I spent, I think, about a year during lockdown getting okay at Photoshop. Turns out it was very useful, because I depend on it heavily for my art business, which I'm getting to now. So unfortunately, I have to give you a science lesson, as depicted by the photo in the top right. That was a genuine candid photo of me in the lab. And my art is based on molecular structures. So you might recognize molecules on the top left. That's kind of the long-handed way of drawing them out. You have C's, which represent carbon atoms, H's, which represent hydrogen atoms. Lines between them mean they're bonded together. And then at the ends of the arrows, you can see some zigzags and hexagons. And that's basically just, scientists are lazy. We like to get everything written down as quickly as possible. So we abbreviate our molecular structures in lines, essentially. And so, as you can hopefully tell from the long-winded versions above, where the lines meet, that's where a carbon atom would be. And then we kind of just imagine that the hydrogen atoms are there, and that's good enough. And what this does is it means that drawing more complex molecular structures is a lot easier. So you can see down on the bottom right of the screen, that is, I think, chlorophyll. And if we'd have drawn that out in the long-winded way, you'd barely be able to see the structure, and it'd be very hard to tell. So yeah, scientifically, this is where my art is based from. So beginning molecular arts, the pun is very much intended. And this was another, I think this is during the second or third lockdown, I started doing this. But my family, I was doing the photorealism stuff, and my family were like, oh, why don't you try and come up with an original idea? You've got time, just have a go. And so I thought, okay, why not? So on the top left, you can actually see the original kind of scribble I made on my test page in my sketchbook for this whole idea. I find this very cool to look back on now that I still have it, and this has kind of become something. So the basis of my art is taking the molecular structures found within the thing I'm drawing and using a kind of pointillism style technique to create that overall image. So it's best to explain this with an example. On the screen, you can see my original bee drawing. And basically what I did was take the molecules of glucose, fructose, gluconic acid, and hydrogen peroxide, draw those out, layer them up, and use different pen thicknesses to try and create the overall image of a bee. I did not think this would work out at all. This was just a stab in the dark. And the finished picture is on the bottom, which came out a lot better than I had expected. It actually came out so well that my parents demanded it for their wall, and it has lived there ever since, which was quite nice because, to be honest, most of my art had lived like under my bed in a folder for the rest, like for my whole life. So this went kind of okay. So well that I forgot about it for about a year and a half, until the following year when I was coming towards the end of my master's. And at the end of my master's, I was thinking, oh, further education, I want to do a PhD. And I was recommended by people that I had known to start a Twitter account for finding PhD positions. However, I hadn't really got any chemistry things to post on my Twitter account. So I drew this molecule rose, which is made out of the scent molecules found in roses. And I thought, well, so I don't look like a spam account, let's post this on Twitter. So I didn't really think anything of it at the time. Oh, going the wrong way. At the time, I had 9 followers, so no one was really going to see it. It was just to prove that I wasn't like a creepy bot. And it kind of went mini viral within the science community, and it got a lot of attention considering I had about 9 followers at the time. And people were actually asking me to sell prints of my art, like they wanted to pay to buy my art, which was the most surreal experience for me, that someone would actually spend money on my art. And then I got picked up by this science magazine, and they wrote a article for their website about my art. And this also attracted more attention to my art, and more people were asking me to sell it. And I was like, wow, this is crazy. So I decided to do something about it. Luckily, at this point, I just finished my master's degree, so I had a lot of free time. I drew another 2 molecule pictures. So at the top right, you can see an orange and a leaf, and they are drawn using the same technique. And I set about opening up my art shop. And this taught me many, many skills. Bearing in mind, as you've seen earlier, I've never taken a business class. I've never taken an art class at school. So it was a very steep learning curve. I had to teach myself a lot. And I've listed those things, or like the ones I can think of, on the bottom left of the screen. But 2 I'll point out: art photography. That was a whole new realm of chaos. I thought it was just as simple as take photo of picture, send that off to the printers, it comes back, and it's how you're wanting it to look like. No. If you're taking photos of art and you've got white on it, you have to Photoshop it to get the white back. Otherwise, you print off this weird like grey blue color. So that's where all the Photoshopping came in very handy, because I had to try and make the colors on my pictures as accurate as possible. Also, social media. If you're ever thinking of starting an art business, or any business really, the advertising power of social media is actually wild. None of my business would have happened if it hadn't been for social media. And it's definitely, I'm still learning how to use social media properly to advertise my designs. And so I've had to learn a lot in that respect as well. Shortly after I started my art business, I started my PhD at Cambridge. So yeah, that on the left, that's me on my first day when I moved in. And yes, I'm studying both chemistry and physics, and I work in an area called optoelectronics. And for me, I basically design and make molecules that absorb and emit light. And these are used in TV screens and phone screens and solar panels, that kind of thing. And this has been the best experience ever. I get to travel all over the world to talk about science and meet new people. And I get to research super cool state-of-the-art technologies. And I've met some of the nicest people that hopefully I'll be in contact with for a long time. However, it goes without saying, it's pretty challenging keeping up with an art business while you're doing your PhD. I've put on a general daily schedule, which is accurate-ish. It changes quite a lot day by day, but as you can see, I have to do art stuff in the morning before I go and do my PhD, and in the evening when I get back. And I try and keep a strict-ish 9 till 5 with my PhD. I do cut out my lunch break, which is a controversial opinion, but it gives me more time on either end of the day.
Stay on the mic.
As you mentioned, you are very stressful with your PhDs while you're doing art. Have you ever thought of giving up, like due to your academic pressure?
No, I haven't yet. I'm quite strong-willed in that I wouldn't want to give this up for my PhD. I think the best way to do this is, as I mentioned earlier, is to keep my PhD a strict 9 till 5. It's quite easy for PhDs to kind of become a whole day, every day thing. And going into my PhD, I knew I didn't want that to be the case. I think that's helped. There are times though when it does get quite overwhelming and I have a lot going on at once, but I like to be busy, so I enjoy it. And I'm really happy that I still get to do both, because I didn't think that would be anything possible.
Thank you. Thank you, Lucy.
Thank you.
Thank you so much. Are there any questions for Veronica? Veronica is also here.
Hi, Veronica. I was wondering, what software are you using for your 3D animations?
Yeah, of course. I use Maxon Cinema 4D. It's similar to Autodesk, and it's similar to Blender. So you've probably heard of, well, you may have heard of Blender, which is like an open source 3D program. It's kind of similar in scope to what it can do.
Thank you. I would like to thank the speakers, both Lucy and Veronica. Really, really wonderful talks. I'm sure everyone must have enjoyed your journeys and will take good learning from both of your talks. So thank you so much for your time and sharing your experiences with us today. Thank you. Okay, so I would now like to present prizes. There are two categories that we have had over the past 3 years: best artwork under 16 and over 16 years. So this year, drum roll, artwork for under 16 goes to Zimo Van from Tsinghua International School, Daizeng Lake, China. And the next category for 16 plus prize winner, drum roll, it goes to Evie Brown, Saffron Walden County High School. All right, so they both receive certificates and 50-pound vouchers so they can buy art supplies and hope they continue their art journey. So moving on, I would now like to share more information about the PDB Art project, where you can find more information. It's all on the website. If you go on to pdb.org/art, and you can see how we started. There is a nice film that can tell you about, how you create art, what all is involved. And if you have any questions, you can always come back to us and we will answer. So if you see, there is a drop-down menu and we have created a lot of sections where you can see past artworks and exhibitions we have in the media, where we show calendars. There are lots of resources, and we also collect feedbacks, and you can see who all we collaborate with. So this year's exhibition, although we have a mini exhibition outside, the exhibition is online. You can use the QR code to visit the online exhibition on Art Steps to see the beautiful artworks which the students have created this year. The calendars are also here. The new calendar, which is 2025 calendar, is here outside, so people on campus can take free copies and distribute them. And if you want to download it, it's available through the link which is over here, and we will share this later. Then, like I said, there are lots of resources if you want to learn more about PDBE. We have lots of activities and tutorials online, and we also highlight the artworks through featured structure articles every month. These are based on the artworks that are present in the calendar, which will give you a brief overview of not only the artwork, but also the science and the function of the protein. Next, like Samir mentioned, we have gone broader, European-wide. The art project has gone to Europe, where EMBL has adopted, as part of their 50th anniversary, this program, which is called Unfold Your World. If you want to participate, the deadline has been extended, and submissions are now open until 18th of October. So this is an expansion of the PDB Art, and there are lots of resources, again, over here, where you can see what all is involved. There are inspiration boards you can choose proteins from. So this program is for 14 to 18 years old. And this is across all EMBL member states. As I said, people are invited to submit their artworks, and these will be displayed in the World of Molecular Biology exhibition in Heidelberg and also online. There is an award worth 100 euros for each of the winners from different member states. If you want to find out more, this is the link. This will be shared later on. And I would like to encourage you to go to our social media pages. We have a lot of presence, and we share a lot of stories, not only on our structures that are there in the database, but also your artworks which are created by the students. And finally, I would like to thank our speakers, wonderful speakers, both Veronica and Lucy, for coming here and giving such wonderful talks. Thank you so much. I would like to thank Bryony, especially from Comms, EBI Comms, who has helped me put up this event, and Ajay from training team, who has been a wonderful help in setting up the Zoom webinar and helping me with the event. And the PDB team members, especially Rafael and Gazelle, who have helped me with the calendars and also with the exhibitions. Not to forget other team members. Without their help, I could not have put up the event. So thank you so much. And the schools who are involved, the teachers. Without them, it is not possible to run the project. So thank you so much. And not to forget students. Your artworks are brilliant. What you do is brilliant. Even I can't do it. So keep it up. So I hope this has inspired you even more. And you have enjoyed the exhibition, the opening event. I'm sure you will take a lot from this. So thank you for being involved.